Apiblast, hypoblast, amniotic cavity here, and this is the exocoelomic cavity or the primary yolk sac, which is by the growth of the exocoelomic membrane here. There is a cytotrophoblast and there is syncytiotrophoblast. This plate with two layers is the embryonic plate. Embryo is in the form of a sheet in this time. अभी वो बिल्कुल human shape में नहीं आया. It's like this. This plate is known as embryonic plate, and at this side it is of two layered, so it is known as bilaminar disc or bilaminar plate. Bilaminar embryonic disc in the second week. So what are two in the second week? The disc, epiblast, hypoblast, the trophoblast with cytotrophoblast and syncytiotrophoblast, two cavities, amniotic cavity and exocoelomic cavity, or the primary yolk sac. One point which I must emphasize here are these 3 days of implantation, and secondly, production of a hormone by syncytiotrophoblast. The blastocyst on 8th day is partially embedded, on 10th day completely embedded, and on 12th day, as I've shown you, even the surface defect that is completely closed. And the normal implantation site is the posterior wall of the uterus. Otherwise, the implantation will be abnormal. The syncytiotrophoblast cells, they start producing a hormone where as soon as they are formed, this is known as HCG, which is the human chorionic gonadotrophin, and many of us might be knowing this, which is actually the pregnancy test. This HCG initially comes in the maternal blood and then it passes into maternal urine, and this forms the basis of the positive pregnancy test. If HCG is there, it will give positive pregnancy test, and this is in the second week of development, right? So the bilaminar embryonic disc is this, which is the hypoblast and the apiblast here. The second week going down, and this is the erosion of the endometrial glands and the blood vessels, forming the lacunae in the syncytiotrophoblast, and syncytiotrophoblast is spreading all around. Syncytiotrophoblast is all around, and you can very clearly see two cavities: the amniotic cavity in blue, and the primary yolk sac or exocoelomic cavity is this pink zone, which is lined by exocoelomic membrane here. अच्छा, एक चीज और यहाँ पे हो गई है, and what is this? That you can make out this purple layer now. There's no purple layer at this stage, right? There is just the cytotrophoblast here, and then the cytotrophoblast and the inner layer, which is the membrane of this primary cavity. Now, within this primary yolk sac and the cytotrophoblast, the primary yolk sac wall, which is exocoelomic membrane, and this is the cytotrophoblast, which are in close contact in this stage. Can you make out this difference? The two walls, they are in close contact. The wall of the primary yolk sac and the cytotrophoblast, they are in contact with each other. In the next, they are separated by this purple zone. This purple zone is another connective tissue, which is known as extraembryonic mesoderm. I should write somewhere here this word, the extraembryonic mesoderm, and this is, I should write it here. Extraembryonic mesoderm. We can see from this slide also that this is the hypoblast, the yellow layer. I'll zoom this diagram. This is the epiblast, and this yellow layer is the hypoblast. The hypoblast cells has grown downwards to form the exocoelomic membrane or the membrane of the primary yolk sac or the exocoelomic cavity. And this membrane and the cytotrophoblast, they are in close contact. In the next stage, in between these two membranes, this is the membrane of the primary yolk sac, which is continuous with hypoblast, and this is epiblast, an amniotic cavity here. There is this purple membrane connective tissue, which is known as extra embryonic mesoderm, which will separate the cytotrophoblast from the primary yolk sac. Okay? And within the syncytial trophoblast, the glands and blood vessels, they have thrown the secretion in blood and created small islands or small spaces which contain the blood and the secretion. These are known as the lacunae. Right? So in this slide, you can see that there is, they have specified exocoelomic cavity or the primary yolk sac. The cells migrate from hypoblast and from thin exocoelomic membrane, and this membrane surrounds the blastocyst cavity and form the exocoelomic cavity, which is actually the primary yolk sac. ठीक है जी. And then the next stage is that now what is happening is before I show you this happening, I should show you this slide again, and the extra embryonic mesoderm, which is this purple zone. Within this extra embryonic mesoderm, the next diagram is a little confusing. In the next slide, I'm going to show you here. This cavity splits by the appearance of small spaces. Small spaces appear within this cavity, and it will cut this mesoderm into two parts. Are you getting this point? There will be small cavities within this pink zone or the purple zone, and they will join up to form a large cavity like this, and which will cut this purple zone into two layers, an outer layer and an inner layer. The inner layer is the inner part of the extraembryonic mesoderm, and the outer layer is the outer part of the extraembryonic mesoderm, and in between there will be a space, and that space is the future chorionic cavity. That is the future chorionic cavity, which you can see in this diagram, that this is the amniotic cavity. This is the, I'll show you the video also again, and so that you can see the bilaminar disc, which epiblast, hypoblast, amniotic cavity, and primary yolk sac. And this, they have changed the color of that purple zone. This is light purple or little whitish. This is the inner layer of extraembryonic mesoderm, and this is the outer layer of extraembryonic mesoderm. And in between, there is a cavity which is known as the chorionic cavity. This is known as chorionic cavity, and the chorionic cavity is surrounded by a membrane, which is the chorionic membrane. You can see this, the chorionic membrane here, which is actually the wall formed by the outer layer of extraembryonic mesoderm, the cytotrophoblast, and syncytial trophoblast. Right? The inner layer of the extraembryonic mesoderm, outer layer of extraembryonic mesoderm, in between chorionic cavity, and this outer layer plus cytotrophoblast, syncytial trophoblast is forming a thick membrane which is known as the chorion, which is forming the wall of the chorionic cavity. Right? So this is about the formation of the chorionic cavity. एंड वन थिंग मोर, अच्छा इसी के अंदर आपको एक और चेंज नजर आ रही है, and that change is within the cytotrophoblast. Can someone point out what is the change in the cytotrophoblast? Can someone point out a change in the cytotrophoblast in this stage? Can someone point out the change? No? कोई चीज नजर आ रही है differently? Can you hear me?
Yes, ma'am, we can hear you. वो slide में इतनी सही नहीं नजर आ रही.
स्लाइड सही नहीं नजर आ रही. नहीं, ये साइटोट्रोफोब्लास्ट पता चल रहा है. ये साइटोट्रोफोब्लास्ट पता चल रहा है, और ये उसमें क्या नजर आ रही है? ये cytotrophoblast, which was a smooth layer, now it is showing you a little growth or proliferation. The cytotrophoblast was a smooth layer, but in certain regions at embryonic pole first, there's a growth of cytotrophoblast, and this growth of cytotrophoblast in the syncytial trophoblast is the beginning of formation of placenta, and it is known as primary villus. The cytotrophoblast growing into the syncytial trophoblast is the formation of primary villus. This is the magnified portion of the cytotrophoblast, syncytial trophoblast. The cytotrophoblast is showing you this proliferation or the growth. This is known as the primary villus or